In this video, we are discussing about the next point, gene interaction. The Mendelian study or Mendelian laws are studied on the basis of pea plant, and these plants show a two genes alleles for one character. In the next study, the phenomenon of this one pair of allele for a trait or phenotype is failed, and that is included under the title gene interaction. The phenomenon of two or more genes affecting the expression of each other in various ways in development of single character of an organism is known as gene interaction. Means one phenotype is controlled by more than two alleles, and not only more than two, but they are interacting, affecting each other. The most of the characters of living organism are controlled, influenced or governed by collaboration of several different genes. The Mendel and other workers assumed that the characters are governed by single gene, but later it was discovered that many characters are governed by two or more genes. Such genes affect the development of concerned characters in various ways. This leads to modification of typical dihybrid ratio, trihybrid ratio, etc. This modification of dihybrid or trihybrid ratio or the interaction in between the genes is true for many phenotypic characters because the characters are hundreds in number and the genes which are lots of in number, means few characters are carried to next generation with help of the lacks of the genes. Hence the one gene pair for one character, this assumption of Mendel is get failed. In previous lecture, in previous video, we are studied about failure of law of dominance. In this, we are studying the next, that is interaction of genes. The gene interaction means coordinated action of non-allelic genes controlling a single phenotypic trait. There are three types of interactions. First one is simple unmodified Mendelian ratios. Second, epistasis, or in another term, modified Mendelian ratios. Third one is quantitative or polygenic, that is continuous variation. For example, height of human being. The typical dihybrid ratio, that is Mendelian ratio, 9:3:3:1. This type of gene interaction produces typical dihybrid ratio of 9:3:3:1 in F2 generation for a single character. Evidently, the concerned character is governed by two genes showing a complete dominance. In case of chickens or fowl, comb shape is governed by two genes, while one gene gives rise to rose comb, another gives rise to pea comb. Each of these two dominant over a single comb. However, when both genes brought together, a new phenotype, a new character is developed in comb, that is walnut. These types or shapes of combs are In this diagram, walnut is developed due to gene for P as well as gene for rose, and the single one is absence of dominant gene for P as well as rose. Hence, the four varieties of combs are developed. This example is studied as worked out by Batson and Punnett. When both dominant genes are present, walnut phenotype appears, and when both recessive alleles are present, single comb appears. Rose and P phenotypes appear due to the presence of different single dominant genes. If P plant having a genotype r capital P and rose having genotype R small p are crossed, the F1 birds showed a walnut comb as it has a dominant allele of both genes P and R. Later on, when the F1 walnut combed birds were inbred together, in F2 generation were appeared walnut, rose, pea and single combed fowls. These types occurred in proportions as 9 walnuts, 3 rose, 3 pea and 1 single. This mode of inheritance of gene for rose and pea does not differ at all from the usual Mendelian scheme. The difference that distinguishes these similar cases from simple dihybrid inheritance and that the F1 does not resemble with parents, and apparently second one is apparently new unusual character walnut result from the interaction between two independently inherited genes, and the other, that is recessive single comb, result from the interaction of their two recessive alleles. These peculiarities are not due to new method of inheritance but simply to the fact or circumstance that both genes involved happen to express themselves in this case. This can be represented diagrammatically as, that is, parents used P and rose, both have a genotype r capital P and capital P R small p. Their gametes represents the gene for both characters. First parent has small r capital P, second one has capital R small p. F1 gate is heterozygous. Gene for dominant both characters is present, but F1 progeny is having different, a novel character, a new character called walnut, and when inbred the F1, this walnut produces a 4 types of gametes. Ratio is same but difference in the F1 phenotype and the representation of its single character. It is also again new. It is again different from the dominant parent. Walnut, which is found in F1, it is also again different. This chart shows a F2 progeny which has 9s2, 3s2, 3s2, 1 ratio, that is 9 cases are having both dominant genes, may be of homozygous or heterozygous, but they produce a walnut comb. another 3 cases produce a rose because they have a dominant gene for the rose comb. Remaining 3 produce P, that is they have a dominant gene for P, and one case is there, it has the both recessive genes, it develop into single. Ratio is same but result is not same. It can be well understood in colorful diagram. Okay, next in this we are studying this interaction of gene showing a non-modification of the Mendelian cross, Mendelian ratio. In next, the modification of Mendelian ratio, that is 9:7, and it is called as complementary interaction of gene. It is observed in sweet pea plant. Two varieties of white flowered plants were seen in sweet pea plant. Each variety bred true and produced white flowers in successive generations. According to Batson and Punnett, when two such white varieties of sweet pea were crossed, the offsprings were found to have a purple colored flowers in F1, but F2 generation gets a different two varieties, that is 9 purple and 7 white. This is again modification of Mendelian ratio 9:3:3:1, where only one character, that is flower color, is involved. It is clear in the above example that for the production of purple flower color, both complementary genes are necessary, that is capital C and capital P. In the absence of either of gene, flowers became white. Thus, it is clear that genes capital C and capital P interact, and presence of both is essential for purple color in flower. These types of genes in which one gene complement the action of other gene constitute a complementary genes or complementary factors. Complementation between two non-allelic genes are essential for production of particular or special phenomenon called as complementary gene. The sweet pea plant produces such a type of flowers. These flowers are purple and white colored. Purple flower has both dominant genes, capital C and capital P, and the white flowers are developed due to small c and small p genes, as well as only capital C and small p, or only capital P and small c, and due to which they cannot develop in presence of single dominant gene color in flower. This example diagrammatically in this chart you can observe, you can study the white flowers having genotype capital C, small p crossed with another white flowered plant having a small c, capital P genotype. Due to such a type of combination, gametes get capital C, small p, and another one is small c, capital P. The F1 generation has all purple color flowers. Genotype capital C, small c, capital P, small p, and this produces this F1 generation, which has a purple colored flowers and heterozygous condition, that is capital C, small c, capital P, small p. If inbred, we get F2 generation, which parents develop a 4 types of gametes and possible 16 combinations develop a 2 varieties. Genotypically, they are showing a Mendelian pattern, but the phenotypic character developed into 2 varieties, that is 9 purple flowered plants versus a 7 white colored flowers. The F2 ratio produced such a 9 and white combination. It can be understood with help of this colorful diagram. You can see the 9 cases which are purple colored in this chart. That 9 cases has C, that is dominant capital C gene or dominant capital P gene in homo or heterozygous condition develop into walnut, and remaining homo or hetero but single gene is dominant, it cannot develop purple color.